Okay, so hello everybody. I'm Sean Jungbluth. I'm a data scientist at Lawrence Berkeley National Lab working in the KBase group. And today I'm going to be presenting a webinar, a workflow to use metagenomes and for the purposes of discovering novel lineages. With this workflow, we're not going to be going into sort of the core navigation of KBase, like how you find tools, how you navigate around your data. I'll probably touch on those topics a little bit, but for the purposes of this tutorial, we're assuming that you have some baseline familiarity with navigating around inside of KBase. And if you don't, of course, we're happy to assist you in learning how to do that. Okay, so let's dive right in. So as I said, the topic of today's webinar is using metagenomes to discover novel microbial lineages in KBase. Full screen here. And this tutorial guides the users through the process of metagenome assembly and eventually obtaining high quality genomes using a suite of tools. The focus here is again on identifying novel lineages of life. Just as a quick overview of KBase before we get into the details of the narrative I've constructed here, I'm just going to read this verbatim pretty much, but you can all kind of follow along. KBase hosts a suite of tools for metabolic modeling and comparative phylogenomics of microbial genomes. But prior to the application of any of these microbial genomics tools, first we must obtain high quality genomes via cultivation or genome extraction from metagenome assembly. KBase has workflows that provide users the ability to construct high quality genomes from both isolates and mixed populations. And again, the focus today is on mixed populations. This webinar will highlight the application of bioinformatic tools for metagenome assembly and genome binning with an emphasis on the recovering of novel lineages from a deep subseafloor environment. In particular, this narrative highlights the effective use of employing multiple genome binners and a bin optimization approach. And the data that we're presenting here today are the result of processing two public deep subseafloor metagenomes that originated from the Berkeley lab at the Hawaii Institute of Marine Biology. So the main lessons from this narrative, we hope that users will learn how to perform quality control of their read libraries. Our goal is to convey how to measure taxonomic population structure with environmental shotgun reads. We're going to have a quick assessment of how we assemble metagenomes and subsequently compare their assembled quality. With the assembled metagenomes, we're going to learn how we can identify closely related assembled public metagenomes in the EBI database. Afterwards, we'll highlight how we use metagenome binning, a mixed approach to metagenomic binning to produce metagenome assembled genomes, or MAGs as they're often called. We'll use an optimized approach there, of course. And once we have identified our genome bins, we'll assign taxonomy to the MAG lineages using a few different tools. We'll assess MAG quality and extract individual genome assembly objects. And finally, we'll conclude with teaching users you all how to annotate genes to obtain KBase genomes, which can be used in downstream analyses that include things like placing MAGs and phylogenetic trees. Here are a few links to related narratives that touch on very similar topics to what I'm presenting here. For a related narrative with extra emphasis on different metagenome assembly options, you can see the narrative here. This one was constructed by Dylan Shivian, and he's a colleague at LBNL, and it's a nice example of the suite of metagenomic assembly tools we have and how to use those effectively on system. And then once you've generated your high quality genomes, you can also check out these two narratives. This first one is by Dylan Shivian, focuses on doing comparative phylogenomics. You can click the link there if you want to check that one out. And then this last one is focused on metabolic reconstruction and community interaction modeling. And I believe that one was constructed by our colleague, Jose Argon. Okay, moving forward, this is a kind of a summary, a table of contents, if you will, of the narrative sections that we have here. There's 9 different sections in total. The first will focus on read import, quality analysis, and end pairing. Then we'll transition into taxonomic classification of raw reads. From there, we'll jump into metagenome assembly and performing metagenome assembly comparisons. We will then take our assembled metagenome and bin contigs from it. After we've performed genome binning using several tools, we'll use a tool called DOS tool to optimize the contents of our genome bins. After DOS tool is finished, we'll assign bin taxonomy and quality assignment to those megs. And we move into the next section where we're going to extract the individual assemblies by their domain, dividing things up into bacteria and archaea. From there, that allows us to annotate genomes for bacteria and archaea accordingly, and that transitions to our last section where we build a tree and find some of the closest relatives. And each of these links are clickable, so you can skip to various sections in the narrative if you feel the urge. A quick word on timing, and this is text that I stole straight out of one of Dylan's narratives because it's important to convey here. So while KBase boasts faster processing and run times for many apps compared to our competitors, keep in mind that large data sets do take time to analyze. And the topic of today's webinar is metagenomes, and those are often relatively large data sets. So queue times for metagenome data sets can be lengthy, especially during high periods of traffic on the servers. But once you're through the queue, hopefully you enjoy relatively quick run times on the span of hours or days for complex algorithms that could possibly take months on other computational hardware. Okay, just a quick description of the apps that we're going to be using today. The first one is called FastQ. This is a tool that allows users to check the quality of their raw sequence data generated through high throughput sequencing pipelines. And the results produced, they differentiate your sequence data, the quality of your sequence data, such that it's colored to highlight the high quality and low quality regions. It's a useful QC tool. We'll use FastQ Join to join paired end sequencing data, which produces a single end library, which is ideal for the taxonomic classification of raw reads. After we've paired our reads, we'll use Kaiju, which is a tool to generate fast and sensitive taxonomic classification for metagenomic reads by comparing sequences to databases of known microbial proteins. That's a tool that works well, and it's very similar to Gotcha2, which employs a similar approach to produce taxonomic classifications based on unique nucleotide signatures. So we'll take a look at both of those. Then we get into our DNA assembler. We're going to be using Megahit, which assembles metagenomic reads using this tool. After that, we will take a look at the, we'll be running two assemblies, and we'll perform a comparison of the contig distributions using this app compares assembled contig distributions. And this allows users to view some of the statistics of their assemblies, which is useful for selecting the best assembly. From there, we'll use Shmash. I think that's the correct pronunciation, Shmash, to allow users to search against EBI metagenomic data for closest matches. And just a note, this app is in beta. That's highlighted right here. And just a quick word about beta apps. Beta apps are not quite yet in production, and therefore there are likely a few bugs associated with those. And so just something to keep in mind as we're moving through this narrative, that some apps are in various developmental states. Okay, with that caveat out of the way, we will move on to using MaxBin2, which uses a nucleotide composition information, source strain abundance, and phylogenetic marker genes to perform binning through an expectation maximization algorithm. We'll also use another genome binner called CONCOCT, which groups assembled metagenome contigs into bins using depth of coverage and nucleotide composition. This is a beta app. And after we've run the genome binners using a couple different techniques, we'll feed those into DOS tool, which optimizes the genome bins using a dereplication, aggregation, and scoring strategy. Again, this is a beta app. After we've run DOS tool, we'll take this tool GTDB TK classify, which assigns taxonomy to bacteria and archaeal genomes based on the genome taxonomy database. We'll also take those same genomes that were produced by DOS tool, and we'll assess the genome quality with CheckM. CheckM provides a set of tools for assessing the quality of genomes or metagenomes, and it produces robust estimates of genome completeness and contamination. After assessing the quality with CheckM, we'll use this app called extract bins as assemblies from bin contigs, which extracts our bin contig data set as an assembly object. And I'll explain the morphing of objects as we go here. Afterwards, we'll run the annotate microbial assembly app, which annotates a bacterial or archaeal assembly using the RAST pipeline, which is the rapid annotations using subsystems technology pipeline. And then getting into our last two apps here, we have the insert set of genomes into a species tree, which constructs a phylogenetic tree combining the genome set provided by the user with a set of closely related genomes from the KBase list of species. And the last app, build microbial species tree, which builds microbial genome, sorry, I should say species tree, optionally including a tree skeleton and phylum exemplars. And final note on that one, that is also a beta app. Okay, with the introduction to all the KBase apps out of the way, just a quick highlight about what kind of samples we're going to be working with today. I'm not going to read this all verbatim, but the samples here were selected because they contain microbes that are pretty highly unique, and they were extracted from deep below the volcanic subseafloor. So deep below the ocean surface, around 250 meters of sediment thickness, fluids actually circulate through the deep ocean crust. And two rare samples were collected from this environment and used for environmental metagenome sequencing and genome binning to reveal novel lineages from the bacterial and archaeal domains of life. And this work has been published in multiple studies using different tools, so they may not match completely with the years-long previous analyses that were performed. The results that we have today may not match previous results, but it's important to know that these data have been published on before. This narrative is meant to reenact the core analyses needed to take raw reads and construct genome bins representing novel lineages of life. And this next paragraph is a little bit technical, so instead of reading it, we'll kind of skip down to this nice little infographic here that highlights some of the cool aspects of these samples we're working on. So the drill ship would have gone out to the ocean and drilled a hole in the subseafloor in some previous year before we started sampling. Also in years before we started sampling, the ocean drilling ship would have placed one of these sort of undersea wells inside the seafloor, and that seals up the rock below here so that it can kind of return to its native state. And then in follow-up studies, we essentially use tools like the deep submersible vehicle Alvin and the remote operated vehicle Jason to go down 2,600 meters to the bottom of the seafloor. We plug into this well here, and then we physically extract fluids and the microbes residing in the fluids from deep down below. It's a very unique environment, and with that, we were able to recover some pretty cool and novel lineages of life. Okay, this is a little bit of a blurb about the protocols that were used for DNA extraction and environmental DNA sequencing. And the technical speak is all located there in that paragraph, but to speed things up, I'm going to highlight one of the takeaways from reading these results. And specifically, two libraries here were generated. One was from this location called 1362A, I'll just call it A from now on, and one from location 1362B, which I'll call B from now on. From reading these methods, there's one very important thing to note about the amount of starting input DNA, which was much larger in the sample collected from A. The amount of starting material dictated what methods were used at the JGI to perform DNA sequencing, and ultimately these samples fell into two different bins of quality based on the amount of input DNA that was present. As a result, the libraries are different in that the library from location A was generated using unamplified environmental DNA, and library B was generated from library, library B was generated from a sample that required whole genome amplification to increase the amount of starting DNA material. Now we know that WGA, whole genome amplification, is known to introduce additional biases into the library prepared for sequencing, and as a result, the read coverage information for library B used in downstream analyses will be lesser quality compared to the equivalent information for library A. And these are important points to keep in mind when we're performing genome binning, because the best tools and the results of using these tools is going to be contingent upon the quality of our input data, which can be prepared in different ways. And so I think an important takeaway here is to make sure you understand how your sequencing data was prepared, and you ensure that you have understood all of the caveats of what you need to do accordingly with your analysis. Okay, with all of that, we are now ready to get into the analysis steps. So step one here is to import reads, perform quality analysis, and quality filtering and end pairing. This workflow begins with the import of reads from the metagenomes 1362A and B. In this instance, reads were copied directly from another KBase narrative, but the original app used to import these data was called the import FastQ/SRA file as reads from staging area app. Other KBase documentation highlights best practices for importing data, and we can provide links to that so folks can know how to do that easily. After our data is loaded into the narrative, one of the first tools that we use is this FastQC tool. Here in this first step, we will also be joining our raw sequencing data in preparation for short read taxonomic assignment tools like Kaiju and Gotcha. Two notes associated with this step. So these data have already been quality checked and filtered according to standard protocols at the Joint Genome Institute. So FastQC application here, the FastQC application here is mainly for demo purposes only. We're not going to take any actions based upon this information because the JGI already quality trimmed these data. Another note is that the read joining is only for preparation to use the short read taxonomic assignment tools. We will not be using the join reads to perform the metagenome assembly. Okay, let's take a look at our first object. So this is the metagenome from sample A, the data object that was produced upon import. And in this object, we have just shy of 300 million reads. This is a paired end library that was produced with Illumina sequencing. It is a mixed population genome, and we do not necessarily know the mean insert size or standard deviation yet. Those are the highlights from the sample from A. Here's a bit more information if you're interested in extracting the total number of bases, the mean sequencing read length, the number of duplicate reads, etc. There's some nice information to extract there. We'll collapse that panel back down. And now we take a look at our 1362B sample. So this was the second sample, and in this one, there's about half the reads, a little over half, 162 million. This library was prepared in the same way in the form of its paired end Illumina sequencing from a mixed population genome. But again, this sample from B was produced from whole genome amplified material. So that's one caveat to keep in mind here. Okay, let's run our first app. So this is the Assess Read Quality with FastQC application. Let me get the configuration box here first. So if you add the app to your panel, let me just do a quick demo of how I would add this app to the panel if I was, add this app to the narrative if I was going to do this, just so everybody's aware. So I just click the app right there. And so in this case, I would have selected the 1362A reads file and then click this run button right here. So I'll delete this new app pane because we've already run this. And I'll minimize this again. Okay, so that's the configuration, and we can take a quick look at the result here. So this is the report one gets when you run FastQC. You get a lot of information about the quality of your file. You get some nice of your input data. You get some nice basic statistics here at the top, which is semi-redundant with some of the information we've already seen. And perhaps some of the more actionable information are these plots that highlight the per base sequence quality here, where it shows on the span of our 150 base pair reads, starting from the first sequence base to the 150th sequenced base. It highlights the quality on the y-axis here as a function of crossing that molecule. And so this is a very common profile that we see in Illumina sequencing, where the quality is pretty good at first, and then it starts to tail off as we approach the end of the sequenced read. But overall, and consistent with how JGI has already QC'd these data, these are pretty good in my opinion right now. There's no data where the average points are in the red zone or the yellow zone here. So these data have already been QC'd. Important to keep in mind. You get more statistics here, like per tile sequence quality, which I don't use that too often, per sequence quality scores, per base sequence content, which you want to see sort of this nice average over the core, over the full length of your molecule to ensure that there's no nucleotide depletion going on in any of your sequencing steps. And additional information related to the GC content on a per sequence basis. You can see that this one's bimodal, which is interesting unto itself. Yeah, I think that's all I'll say about the results of the FastQC report right there. Happy to discuss that more afterwards if there's questions. And expanding the second box here, this is just the second sample, the sample from location B here. Which we ran identically to how we ran the sample A. And just taking a quick peek at the results here, we see, I would say, mainly a good quality profile. It seems like it tails off a little bit more in the B library here. The last base, the average, is in the yellow zone. So, perhaps more trimming can be performed there, but we're not going to scrutinize that today. Okay, so that's FastQC. So that moves us into the FastQ Join application. So here are the input parameters for FastQ Join. We indicate what our starting raw reads are, and then we give it a name here. I called it .joined, and it actually appends its own underscore join, so it says join twice, but that's okay. And I didn't alter any of the advanced parameters here, but you can get a sense of what the mismatch percentage threshold is, as well as the minimum number of base overlap for pairing. You can play with that if need be. So I ran that app, and now we can take a quick look at the results. So in total, there were about 150 million total reads. That's 150 million forward, 150 million reverse reads. And in total, there were about a little over 100 million that were joined. And so we got about 66% of our read library joined. After performing the joining, we also now have access to these statistics to the tune of average join length and the standard deviation of join length, which could be useful for some downstream applications. So that's the result from performing the FastQ Join. Let's take a look at the equivalent for sample B here. So I ran it with the same parameters. Essentially, I plugged in sample B here and appended this underscore joined at the end of the sample name, the export object name. Didn't change any of the advanced parameters. There they are. And then let's take a look at the result here. So in total, there were roughly about 80 million forward reads, 80 million reverse reads, and looks like this one didn't pair as many. It looks like it was about a 50% success rate, give or take, with a little over 40 million reads having been joined. And again, we get access here to the average and standard deviation of the join length statistics, which could be useful downstream. Okay, that's the result of running FastQ Join. So now we're on to step 2, which is taxonomic classification of raw reads. So we generate a preliminary view of our taxonomic profiles in our samples through an analysis of the raw sequencing reads. In my experience, which is, I wouldn't say extensive with running these short read classification programs, but I always tend to have better success when I run joined reads into these tools. And this maximizes our chances at finding a good database hit, because essentially we have longer length sequences with which we're using to perform our query. Here we're going to be running both Kaiju and Gotcha2 using multiple different reference databases so that a comparison of the results is possible. And one note, I only ran sample 1362B here through the raw read analysis data, but this will be updated soon to include the sample A. There was a snafu with me not having the joined reads in time. So apologies, but I'll update that so you can see very soon how the two samples compare. Okay, Kaiju. Before we take a look at the results, here's a glimpse into how I ran it for the sample of 1362B. So I inputted the joined reads here. It's got the underscore joined twice because I typed it in above when I didn't need to. That's all right though, we know exactly what it is. And then after inputting the read set, I selected that we want all taxonomic levels to be classified and that we want to use the refseq genome taxonomic database, which does not have any eukaryotes. These are default parameters, important parameters to keep in mind when you're running Kaiju. We have a low abundance filter and a subsample per set. And the low abundance filter, what this does is it collapses all the lineages, in this case, that are less than 0.5% in your sample. They will all be collapsed into sort of a lumped low abundance group. And this is a way to preserve the legend on these plots so that you don't get too many colors. And then also the library, because it is so massive and oftentimes there is redundant information in the, for example, 150 million or 300 million reads, we subsample these data before feeding them into Kaiju, and this assists with increasing our run times. And I guess we find that that's okay to do, because if you subsample your data, you tend to do so in an even sense. If you don't want to do any subsampling, you can increase this number to 100 to classify all of your reads. Okay, that's how Kaiju is run. So let's take a quick look at the results. We can start to contextualize here our unique samples now that we're comparing to reference databases. So we ran these samples against refseq, which refseq is somewhat limited in form, that it is restricted to mostly isolate data. And so we're trying to compare our deep subseafloor samples against mainly isolate data and see what we find here. So on the left-hand side, we get some taxonomic calls where we've got a bunch of proteobacteria. That's a popular phylum in the bacterial domain. We have a bunch of Euryarchaeota, an appreciable number of Euryarchaeota, 30 to 35%, I would say. And those, of course, come from the archaeal domain. And so this is a nice kind of snapshot into our samples before we've done any assembly. And we know that we have a combination of bacteria and archaeal domains present. And we can perhaps perform our analyses with that in mind and make them more tailored. So here we can see a phylum level clustering of these data, class level clustering of these data, and this goes all the way down since we selected all taxonomic levels to order, family, genus, and species. And you can see here that as we move down to the species level, we get fewer and fewer lineages classified, and that's likely expected given the novel environment from which we extracted these samples. And so this is, I would say, a semi-unique result in that we don't have a ton of hits. We have a lot of things that were lumped into the tail here, or either unassigned at the species level. If you're interested in downloading any of the Kaiju output, including the plots that are shown here, you can download these files here. Oftentimes users will want to download these and work up their own plots offline, say in R or Python or something to that extent. Okay, so that was the first run of Kaiju. I'm going to collapse this cell back down. Let's open up the second run of Kaiju. And I'll show you how I ran this slightly differently. I used the same input library, I used the same taxonomic levels, but this time I used a different reference database. I used the NCBI BLAST non-redundant database, which also does not include eukaryotes. These two parameters down here were kept the same. And so with this database, it's a little bit more expanded compared to the RefSeq database that we used before. And so with that, we'll potentially get higher numbers of matches to things that have not been cultivated before, candidate phyla, if you will. And so that's exactly what we see. We just taking a look here at the phylum level plot here, we now have additional resolution for some of these groups. We now have this new lineage called Candidatus amini-sonantes, Candidatus bipolar colloda. That's a unique one, and Candidatus bathyarchaeota. And so importantly, we're able to get some of these classifications here because we selected a database that was more representative of our query sample to begin with. So a takeaway here is make sure you think carefully about what is the best reference database for you to use for your data. We also have information for all the other levels because we selected that we wanted to analyze all levels. And so this taxonomic classification extends down all the way to the species level. It's not ultra informative when it says Candidatus bathyarchaeota archaean, but nevertheless, these are the names that are being ascribed at the species level in the NCBI-NR database. Okay, so that's Kaiju. Let's take a look at Gotcha 2. So Gotcha 2 uses, it's essentially a similar approach where we are performing taxonomic classification of raw reads. And here we're inputting the joined reads again, and we're using the Gotcha 2 viral and bacterial database. And in terms of advanced parameters, we didn't touch these at all, but you can tweak things to the tune of having a required minimum coverage, number of minimum absolute reads detected, minimum length, and some scoring mechanism for matches. You can tweak all of those. Taking a look at the results here. Each app is different. You notice that the first results page here, we don't have these plots that we saw in Kaiju, and that's because this app was generated by a different developer. Instead, we have this results table on the front here. And so we can cruise around our results and get a sense of our data that way. Perhaps a better way is looking at this dendrogram plot, which I'll admit that for our case, this isn't going to be probably the most useful. We have some pretty highly unique data, so they may not fit on a tree well. Let's take a look at this chronoplot here. The chronoplot provides some good resolution, I would say, in terms of looking at the bacterial domain. One thing to note here is that archaea are missing, and so since we know from our Kaiju run that we have a lot of archaea in the sample, and we're not seeing any archaea here, I think that this is reason to question some of these gotcha results. And so, just things to keep in mind as you're stepping through your data and running multiple tools that have similar goals, i.e., in this case, classification of raw reads. And so, yeah, you can explore some of these lineages in higher levels of detail. Gotcha 2 is unique in having a viral database that you can use as a reference. And so you can start to explore some of the taxonomic makeup of viruses in your sample with Gotcha. So there's Gotcha2 running against the bacterial and viral database. And I ran Gotcha2 again using a fungal database, which I didn't realize they had until yesterday. And so I was eager to run this library and see what sort of fungi were present. And perhaps not surprisingly, because we had not detected any fungal sequences in there, I got this quote unquote error message here saying that there is no taxonomic classification assignment against the fungi database used here. And so it recommends that I may want to try another database, but in the end, this is probably some bit of confirmation that there's no fungi in our samples from the deep subseafloor, or there's very diverse divergent fungi from the Gotcha2 database. Okay, so there's two different ways that we can run Gotcha2. All right, so now we're at step 3, which is metagenome assembly and comparison. So at this point, we have ensured that our sequencing reads are, Windows jumping around. All good now. So at this point, we've ensured that our sequencing reads are cleaned up and ready for assembly. And here we're using the Megahit assembler using the meta-sensitive parameter set. KBase has a range of metagenome assemblers from Megahit, Metaspades, HIPMER, and IDBA. And for a good demonstration of their execution in the KBase environment, I recommend that you check out this great tutorial from my colleague, Dr. Dylan Shivian, titled Genome Extraction from Shotgun Metagenomes. There's a link right there. After we produce our metagenome assemblies, we'll run this tool shmash to find the closest matches in the EBI metagenome database. Just a quick note here, this narrative is focused on the application of multiple genome binning 2. And so here we're not placing an emphasis on obtaining the best assembly. But very important, it's important that you check this out because for your own work, it may be ideal to produce the most contiguous assembly or the most assembled metagenome possible. Things to inspect. And again, I recommend this tutorial by Dylan, which is very helpful in navigating these challenges. Okay, it takes us to Megahit. So this is the app assemble reads with Megahit, and this is how I ran it. These were the raw reads, so not the joined reads. Those were just for the raw read taxonomic classification purposes. Here we use the unjoined reads, the paired end reads, because those are, there's a lot of information there that the assembler uses. Specifically with the sample from location A, we lost about 33% of the reads during the pairing, and we lost about 50% of the reads for sample B. And so we include all of that information here for the assembler to use. The parameter preset that I selected, this was changed from default. I selected meta-sensitive, but there are other parameter presets that you can select if you have a highly complex sample and you are trying to see if you can get an assembly period. Here I did not change the minimum contig length. It is by default 2,000 base pairs, which seems like a reasonable cutoff for making sure that you have some gene content on your contigs. And then we give it an output name. So this tool ran in, I would say like 8 hours, 12 hours, something like that, just to give you a sense of how long. Actually, I can tell you exactly. 8 hours, 35 minutes here. So for 300 million reads, I would say that this is pretty darn quick, just to give you a sense of timing and the power of our equipment here at KBase. So when you run metagenome assemblies on KBase, it automatically runs this QAST tool for you, which is the quality assessment tool for genome assemblies. And from running QAST, we get a variety of statistics that are often used and are highly informative for selecting the most ideal metagenome assembly. Here we get a sense of the number of contigs that were produced in total. We get a sense of the number of contigs that were produced at different ranges. Specifically here, one contig was above 1 million base pairs, which is pretty large for a metagenome assembly. 221 contigs were above 100,000 base pairs, and 3,369 were above 10,000 base pairs. So that's a fairly good assembly. Here we get a sense of how many bases are within the different bins of assembly length. We also get access to statistics like the N50, N75, L50, L75, which can be highly informative, and they give you a sense of the number of, how good your assembly is in terms of the number of contigs that are required to represent 50% of your assembly. And in this case, it was 30,000, or no, sorry, I have these reversed. The N50 is the length of the contig for which, above which 50% of your assembly is produced. And the L50 is the minimum number of contigs. Oh, here we go. I'll just use the tooltips. The minimum number of contigs that produce half of the bases in the assembly. So good statistics for you to effectively sense if your assemblies are working or not. We also get a sense of mismatches, which ideally here are 0. Okay, that was the result from assembling the sample from location A. Taking a look at the location B, which we ran in similar form here. We just imported a different library, the same parameter set here, the same threshold for minimum contig length, and a slightly different name. And in the result pane here, we get a sense of the quas statistics again. And here we did pretty good. We had 3 contigs this time that were above 1 million base pairs, 176 above 100,000, about the same number above 10,000. And so, yeah, these assemblies are fairly comparable. We also have 0 mismatches in both assemblies. Okay, so that's taking a look at some of the statistics separately. We also have access to this application here called Compare Assembled Contig Distributions. I'll show you how I ran that. So the object that was produced from the metagenome assembly is this first one here, and the object produced from performing the assembly on B, on sample B is located here. It's a very simple input. And here's a quick look at the results. So this is more of a side-by-side comparison of your different assemblies. And I don't think we have any sort of a priori expectations for which assembly should be better or worse here. This is just sort of for demo purposes. But here we highlight the sum of contig lengths on the y-axis and the rank of the sorted contig order from longest to shortest here. And so what this highlights is essentially that the assembly from location A, it seems to be more complete, and it seems to be, it seems that there are larger numbers of longer size contigs in sample A compared to sample B. However, sample B does seem to be more assembled as we kind of eclipse the height of the blue line here. Here's, I think, similar information, just sort of permuted in different ways, so you can analyze it as your brain likes to best approach things. And here we have a nice summary, somewhat redundant with some of the quas statistics, but a nice tabular and graphical summary of your contig lengths and statistics. So we have some nice highlighting there. And it's pretty neat to see that this color key of, from best to worst, which, really helps guide the eye toward what are the superior components of your individual assemblies. Okay, that's compare contig distributions. So now we're going to use containment minimum hash, SMASH. So containment minimum hash, this allows us to identify closely related metagenomes in the public database. Here I've input the sample from 1362A, the assembly from 1362A, location A, and I use the reference database, the only reference database that we currently have right now, which is the EBI Metagenomes. And I kept this, this number at the default 10 number of closest matches. Okay, taking a look at the result here. Oh, importantly, this is a tool that's in beta. And so if you're using this and you see bugs, please let us know so that we can squash those out and improve our system. So here the app results, they give you a sense of what are the closest samples in the EBI metagenome library that are related to our samples. And in this case, we have this assembly ID ERZ529318 here. We can open this link, and this takes us to the ENA page where we can learn a little bit more about this metagenome. This looks like it's a marine sediment metagenome, which is interesting because our samples are from the rock itself. And we can get more information about this sample if we continue clicking through here. Oh, so interestingly, this is actually from the subseafloor in a nearby deep sea well location, and it's also basaltic glass. So that's cool. That's probably a real hit. There's probably a lot of taxonomic overlap there. So cool. That's a very powerful new tool that we have in KBase, and allows you to identify the closest metagenomes in public databases. There's other information here that I'm not going to extensively discuss, but you can play around and let us know if you have questions about any of this information. Just clicking through here. We also provide, it looks like a nice map of where the closest hits, the samples were located. It seems like we only had 2 hits in this case. So that's kind of interesting. Okay, minimize this one. Let's take a look at sample B. Okay, configuration-wise, we ran it in similar form. We inputted the assembly from location B, and we ran it against the EBI metagenome database, and using the same number of expected results. And let's take a look at the results here. So here we are, the name of our input query, and can follow the links here to see what the closest sample for this particular genome is. It looks like it's going to be the same sample, which is perhaps not surprising because this is a fairly unique environment. So there we go. That's kind of a nice ground truthing of two samples collected from the same environment, run in parallel, and we get essentially the same result. So I would feel pretty good about saying that these metagenomes are related. Okay, so that's SMASH. All right, that takes us to step 4, which is binning contigs. So after metagenome assembly, we're ready to cluster contigs in bins that correspond to putative population genomes. For the genome binning, here we're going to use multiple genome binners and prep for a bin optimization approach. 2 notes here. So we've had some recent system updates that have introduced some new bugs that are preventing MetaBat2 from working. We'll fix this ASAP, but for the time being, we can't, I couldn't run the MetaBat2 tool, and so we'll get that fixed up ASAP. And another note is that we're going to be, you see MaxBin and CONCOCT are listed here again multiple times. We're using multiple genome binning tools. We're using genome binning tools multiple times, but we're varying the input parameters. And this approach is going to provide us multiple sets of bin contigs that will optimize at the next step. So I won't actually click run here because I don't think it will be successful. There's a unique challenge with our parallelization libraries that we're working out here. But here are the input parameter sets for MetaBat2. So I would input the metagenome assembly here, and I would input the raw reads. Note that this is not, these are not the joined reads, these are the actual raw reads here. And I would give it an appropriate name here. There's also a threshold for the minimum contig length that you can set. And I'd recommend not going too far below 2,500 unless you really know what you're doing. So we can't take a look at the results, but if that once that runs, it'll produce genome bins for us with MetaBat2. The equivalent parameter sets are plugged in here for the sample B. And so we've got the assembly from sample B, the sample B reads, and an appropriate name for the sample B metadata2 bin contigs. And again, if this had run, we would click result here and I could highlight the number of bins that were produced. That takes us to the second genome binner after highlighting metabat. This is maxbin2, which did successfully run. And so I can show you the results here. The assembly object that we input was sample A here. The sample A reads are here. And we input the a unique name for this particular bin contig object. And unique to this particular webinar, this particular narrative that I'm highlighting here, I started to tinker with some of the advanced parameters inside of this app. In this case, the 107 bacterial marker gene set is default, and so that wasn't changed in this instance, but I did increase this minimum contig length up to 2,500, up from, I believe it's 1,500 by default. So that's one altered parameter set that I put in there. Here we can take a look at the result that is produced. So from running maxbin2 on sample A and using the 107 gene marker set that's tailored for bacteria, we identified 58 total bins. The total number of input contigs was 15,500, give or take, and we binned about 2/3 of it. It's a pretty good binning attempt. We get a sense of how much was unbinned, how many contigs were too short, and some other statistics that could be useful. And if you're interested in downloading the maxbin2 result, you can click it right there, and that provides you a zip file with all of the files that are produced that when running maxbin2 locally. So there's the first way that we've run maxbin2. Let's take a look at how I ran it here. So these are all the same input parameters used for this sample A here. And clicking the advanced menu and taking a look at the marker set used. In this case, I used the 40 bacterial and archaeal marker gene set. So this is a marker gene set that is more geared for picking up archaea, which we know from looking at our Kaiju results, they are a substantial fraction of our raw read data. And so that was perhaps an insightful result that we are able to employ here to get a better result. And so we selected the 40 gene marker set, and then I indicated that this was the 40 markers used to produce this bin contig object. The identical operations were run for this sample B. So your sample B with the appropriate parameters plugged in. Here we used at first the 107 bacterial marker gene set, and we set the minimum contig length at 2,500 and named it appropriately. Here I didn't highlight the number of bins from that last step. So the number of bins, when we switched to using the 40 gene marker set, it went up. And so that is perhaps expected, again, because now we're trying more, we're making a better effort to bin archaea. And so indeed, we had 71 bins, which is up from 58. And so notably, that is, we had to use parameters that were not the default parameters for these tools. And so as you're working through your own data, make sure that you are always thinking about how to tailor these tools for your particular analysis. It's key to the development of the highest level insights from your data. And so we have more bins here. That's very cool. I won't discuss all the rest of the statistics, but they're there for you to review should you want to explore those. Okay, back to sample B. So this was the first attempt with sample B. We got 47 bins using the bacterial focused marker set, and with sample B using the sort of broader bacterial and archaea focused marker set, we've got 63 bins. So another improved result from using non-default parameters. So cool. It's great to see that we're recovering higher numbers of archaea in those samples from performing a sort of tailored investigation into our samples. Okay, that brings us to the third genome binner that we have on system. This is a new one, CONCOCT, which is still in beta also. So if you're running it and have challenges with the tool, please let us know and we'll get those bugs fixed right up. And as I'm talking about the beta app, let me just highlight real quick how we locate beta apps on system, because it's important for users to be able to navigate. So I'm going to type concoct here, and you see that no results are showing up, and that's because this app's in beta, and right now we're looking at the released versions of apps only. So we have to toggle into beta mode, as we call it here, and this expands our access to other applications in our catalog that are in more of a developmental state. And so here you can then find concoct, click it, and then add it into your narrative. That's how you find beta apps. Just thought I'd do a quick segue on that, since that can be a challenge. Okay, we're going to collapse this pane back down. There we go. Okay, concoct. So we input these parameters in similar format to how we input them for maxbin and metabat above. We have the assembly and the reads. Again, we're using the raw reads, not the join reads, and we give it an appropriate name here. Here in the advanced parameters, there's a little bit more options. There are a few more options provided to the users here. Specifically, we provide multiple different read mapping tools for you to perform your read mapping at various stringencies, and that allows you to generate coverage estimates that are going to be a little bit different across the different tools. Here you can also tune other things to the tune of the minimum contig length, the contig split size, which is a bit complicated, but during the course of binning, if contigs are too long, they're sort of temporarily sliced into smaller chunks so that there's not too much weighting towards the very, very long contigs. It's technical, but that's a cool feature of concoct. You can also tune the contig split overlap here, which I don't really play with very much. You can play with the size of the k-mers used for tetranucleotide frequency clustering. You can also change the number of clusters and iterations for this algorithm that's used, VGMM, which is the exact definition of which evades me right now. And then the last options that you have provided to you, you can use certain amounts of the PCA space. Here, the default is 90%. You can skip the coverage normalization step if you're interested in doing that. That might be useful if you have sort of unique characteristics in your read coverage data. And you can also skip using total coverage altogether if you want to bin just using nucleotide signatures alone. So a little bit more flexibility within concoct provided to users here. Here we inputted an appropriate name for the result. And let's take a quick look at what we got. So running sample A, we got 88 bins total. And this was the, I think, largest number of bins we've obtained so far from a single sample. Maybe I'm misremembering, but that's a fairly high number. So that's pretty cool. Here, I guess I should note that one of the ways that I ran concoct differentially here is that in this instance, I used bowtie2 as the read mapper, specifically the bowtie2 default read mapping mode. And in this next run with concoct, I changed the read mapper. So these are the same input. And here I used bowtie2 running it in very sensitive mode, which I guess my aim here was to see if we would get different results from performing some differential read mapping. And specifically, I guess I'm interested in seeing if this improved the quality of our genome bins in the end. The rest of the parameters are all the same. And this was the third flavor of binning with concoct, which employed this, this is again sample A here, And this particular run employed BBMap run in fast mode. So that's a different read mapping solution that we offer internally here. Taking a quick look at the results. So we got 90 bins with BBMap, with bowtie 2 in sensitive mode. We got 90 bins also there. And then this one with was 88 bins. So there are some small differences there, just using read mappers. That's perhaps something that you will want to explore with your own data. Here is essentially the parallel running of CONCOCT in these 3 different ways for sample B. So we have sample B with the appropriate parameters plugged in here. Here I did bowtie 2 default. We get a count of the number of bins here. We had 68 bins total here. There's that one. This was sample B with bowtie 2 in very sensitive mode. Here we had 70 bins, so a few additional. And with BBMap, which we ran like so, we obtained, looks like 71 bins. And so there you go. So with sample B, the sample that has, that was prepared using whole genome amplification techniques, you see that varying our read mappers, which is getting at a coverage estimate, gives us a different number of bins resulting from these tools. And so these are important things to keep in mind as you're moving through these tools with your own data. Okay, that takes us to step 5, the bin optimization step. So here we're employing a bin optimization approach using a tool called DOS-Tool. DOS-Tool, it uses dereplication, aggregation, and scoring to optimize the expected single copy marker genes within the contigs. And so essentially that is taking a look at the gene content in your contigs and shuffling those around so that genes we think should appear only once in a genome, that they actually do so. 2 notes here. So running DOS-Tool, we automatically reduce the size of our input bin contigs object because low completeness and high contamination bins are expunged using this tool. So if during the process of binning with Metabat, MaxBin or CONCOCT, you have a bunch of bins that are, say, less than 50% complete or partially complete or highly contaminated, DOS-Tool will, if it can't fix those bins and make them high quality bins, then it will purge those from your bin config object. Another note is that DOS-Tool will not always be able to optimize your input genome bins. There's some instances where an optimized set cannot be identified, and so the program will return an error message essentially saying no bin config object has been generated. And essentially that's not an error, it's just a statement that DOS-Tool is not going to be an effective tool for your workflow. Hopefully those instances are minimized. I'll be eager to see where those crop up. Okay, DOS-Tool. So this is how we ran DOS-Tool. And this one completed just in time before the webinar. Yay. So this one was run for sample A. We input the metagenome assembly here at the top. And then we had 5 different contig sets to select from. We had we didn't have Metabat. We had these 2 from MaxBin, the 40 marker and the 107 marker sets that were used. And then we had CONCOCT that was run in 3 different ways with the read mapper, 3 different read mappers. Here to run DOS-Tool, we're using the gene identification tool Diamond, which is what I've selected by default, in no part because it's faster. And your other option here is Blast. If you, excuse me, if you run DOS-Tool offline, you also have the option of using usearch for your gene identification tool here, but because of licensing restrictions, we cannot include usearch in this DOS-Tool implementation. Here I give the bin contig objects an appropriate name, relatively simple name. And after running that app, which runs fairly quickly because we're not really using any of the raw read information anymore, we get this result. So in total, we have 32 bins. Notably though, these are all bins that are pretty high quality. DOS tool will define it in ways that are different than programs like CheckM, but it seems like all of the genomes that are retained at this stage have a marker gene completeness of at least about 60% based on DOS tool standards. Okay, so stepping through this, this plot here, I'm gonna increase the size of my window. Hopefully, so this is a little bit more legible. Still barely legible, so I'll go through this. On the y-axis here, we have the number of bins, which are colorized according to this key here. On the farthest left, we have max bin 2 with the 40 markers. This is all for sample A again. The second one over, we have max bin 2 with the 107 markers. Here we have CONCOCT with default bowtie 2 parameters. Here we have CONCOCT with the very sensitive bowtie 2 parameters. And here we have CONCOCT with the BBMAP parameters. And ultimately, when this input was fed into DOS tool, it performed its optimization and approach to increase the number of high quality genome bins. So based on DOS tool's assessment of single copy gene completeness, we have, it looks like about 36 or 37 genomes that are above 90% completeness, which is higher than DOS tool's assessment for the rest. We also have at least as large a cohort of genomes above 80% complete, and at least as similar number greater than 70% complete. And so cool, you know, in this case, I think this is evidence that DOS tool perhaps did a good job at optimizing our contig bin content and provided us a superior result than the input from these different binners. There's other information related here that you can explore, such as the actual scoring scale used by DOS tool internally, and you can get a sense of how the different bins stack up in that respect. But overall, I think this plot highlights our good result. Okay, so that cut our number of bins down to 32 in the case of sample A. Let's take a look at sample B. Okay, we ran this essentially in the same way that we did the last sample. We used the input assembly here and the 5 different bin contig objects. We used the same gene identification tool and gave it an appropriate name. Oh, I didn't expand this advanced box in the last time, so I'll do that here and explain that here. So there's other parameters you can tinker with if you are feeling brave and perhaps some level of mastery with DOS tool. You can move the score threshold, the duplicate penalty, and the megabin penalty around. And I don't have much experience doing that, but this is something you can explore with your own data. And you can also choose to write the bin evaluations and plots or not. And these are just produced by default, since that seems pretty easy and useful. Okay, so those were the parameters. Taking a look at the results for sample B. So in total here, we had 23 quote unquote high quality genome bins based on DOS tools assessment. Going from left to right here, I'm going to shrink my browser one level. Okay, there we go. Going from left to right here, we have the max bin with 40 markers, the max bin with 107 markers, which look at that difference in result. That's astonishing. You know, we used the non-default parameters and we obtained almost 3 times the number of high quality bins. That's a pretty notable difference. The results for CONCOCT look pretty consistent. Looks like bowtie 2 in the 2 different flavors, and BBMAP all gave the same number of bins. And it's pretty cool to see that DOS tool had the same results for sample B that it did for sample A, where it took these input bin contig objects, and it produced an optimized result with a larger number of high quality genomes. And scrolling over so you can see the single copy marker gene contiguous scale there. So cool, DOS tool is making better genome bins in the case of both sample A and sample B here. Okay. So that brings us to step 6. So bin taxonomy assessment. At this, we are using a powerful new tool called GTDB-TK to provide taxonomic assignments for bacterial and archaeal genomes based on the Genome Taxonomy database. In addition, at this step, we're using a popular tool called CheckM to assign genome bin completeness and contamination scores. And a few notes as you're working through this webinar narrative. The results from the GTDB-TK tool, we're going to use those to help divide our bacterial and archaeal bins into separate groups, which will facilitate their downstream processing. Another note, in case folks are not familiar with the power of GTDB-TK, the taxonomic assignments provided by this tool, they really do a lot to help assess the novelty of your genome bins. It's a safe bet that if they're not in the database that this tool uses, then you have something that is novel. And then the final note was just remember that DOS tool, it reduced the size of our bin contig objects. So now we're running GTDB-TK and CheckM on genomes that are relatively quite good. DOS tool, you know, it's optimized the gene content, and so these things already should be relatively complete and free of contamination. And at this point, you might want to just make sure that the bins that DOS tool pushed aside are indeed not worthy of your time. So just keep that in mind, that we're, you know, kind of narrowing in on different aspects of our data set over time. Okay, GTDB-TK, let's take a look. So the input parameters for this app are very straightforward. We take the DOS tool bin contigs object that we produced, and I didn't alter this advanced parameter, but this is the minimum alignment % required, set at 10. And this is the result. So the topic of this webinar is exploring novel lineages of life. And I should say that this data, the data that I'm presenting here, the genome bins that were produced, these have been published for about 3 years now, and they have been swooped up and made a part of the Genome Taxonomy database. And so with that, our closest hits are probably going to be the exact genomes that we are finding here today. So I'm going to have to do a little bit of hand waviness, but the point is, when we're finding that our closest hits are related to these things called JDFR. These are the genomes that I've published previously from this environment. And there's an incredible wealth of information to navigate here, but the taxonomy, the taxonomic information provided by GTDB, it's highly informative. And in this case, in the case of bin 1 here, it tells us that we have a Nitrospirota bug from the bacterial domain, and it's classified down to the order Thermodisulfil vibrionales. And then within here, this genome represents a unique family. And so this particular genome, which has a different name here in our narrative, is actually genome JDFR88. If I was a Benton person, that's what I would say. And so it is its own unique family, unique genre, and unique species. So there's kind of a rough sense of some novel taxonomic diversity there. So we're getting bacteria in our samples. We have some of these groups that are called, that are mostly related to, this is uncultured bacteria, I believe, 3574. This is from a publication by Parks et al., the 8,000 genomes one. So this is, this particular bin is not related to any known cultivars. It's just related to this lineage that we've discovered only from environmental sequence data. So that's potentially novel at the genus level. What other results to glean here? Sometimes we have very straightforward results, like there was a Pseudomonas group E apparently within our samples from the seafloor. And so it's good to see sometimes that we're getting very high levels of taxonomic matches there to, in this case, the Pseudomonas. Here's another one. This is a genome that I published and wrote up as a separate paper. We're getting a closest hit to that particular lineage, which was a new species at the time, and that's what we named it. So this lineage was at the point of publication a new genre and species. Here's an interesting novel taxa that is only classified down to the Linealis order. And so here we are unpopulated in terms of the family, genre, and species levels. And so this would be a candidate microbial genome that is novel at the family level within the Linealis chloroflexi. And yeah, I guess I'm just trying to highlight and contextualize some of the novel lineages aspects here. Here's an interesting one that looks like it's only been classified to the phylum level, UBP7, which is a lineage I'm not familiar with. What other information to highlight here? Within the GTDB results, GTDB TK results, you get a sense of the closest ANI reference, the distance from that reference, the taxonomy of the hit, as well as looks like some other information related to closest hits. All good info that you may want to explore as you're moving through these with your own data. There's multiple ways to check out this table. I'll just jump here to the archaeal side of things real quick before we move on. It looks like we're getting unique archaea within the Archaeoglobi lineage. We have these Canadatus Bathyarchaeota lineages, which if you remember Kaiju with the RefSeq database, it wasn't able to identify those correctly, but the NR database, we got a bit higher resolution. So it's nice to see those confirmed with GTDB. Yeah, just kind of other archaea that we have, that have popped up and that we've published on before. So lots of novel lineages. In these particular samples compared to most samples, it's unlikely that you are working in an environment that's quite as inaccessible as the deep subseafloor rock. And so with that, you may not get as many new lineages, but it's likely that through using tools like this and leveraging them in the right way, that you can explore novelty in your samples in ways that were not previously accessible. Well, we're doing good on time. I think I'll just give a quick hat tip here to the results from sample B, which was run in the same way. And because these two samples were collected from the same-ish environment, ideally we would see mostly similar taxa here. And indeed, that is what we see. There's a lot of overlap between chloroflexi groups here, this bipolar Colloda, and some other groups, Munisonantes, etc. So that's great. We definitely expect overlap, both in the bacterial and the archaeal fraction here. We're seeing Archaeoglobi and some others. Oh, I guess I'll save this one. This is, I'll talk about this one real quick. This is, I think it will stand as a novel phylum that we've discovered from the archaeal domain that we've dubbed Hydrotherm Archaeota. And this was perhaps one of the most novel lineages that we discovered and published in our samples. So using current GTDB databases, we actually get an assignment to the group that we named here. So it's highlighting that the tree of life, our blind spots are filling in. Okay, so that's GTDB TK Classify, which is an ultra powerful tool. Please use it. That takes us to check-in, which a lot of folks may be familiar with. Here is how I've run it for the sample A, the DOS tool contigs. And one tweak that I made to the default parameters was I changed the reference tree to be the full tree instead of a reduced tree. And this takes a bit more time to run because the tree is bigger and we're placing our sequences in a larger tree. But in this case, I was interested in making sure that we didn't lose any information with respect to our archaeal bins, making sure that we had good coverage in our reference tree and that we were assessing the completeness and contamination well. And so we can take a look at the result from CheckM. Here we have a nice marker gene plot. This was made by Patrick Chain, and this gives you a sense of the number of single copy marker genes, missing marker genes that are present and thought to be indicative of strain heterogeneity, as well as marker genes that are present and thought to be indicative of contamination. And these are concepts that are fairly common when discussing genome bins, and so I encourage you to check out the CheckM paper if you'd like to take a deeper dive in this. But overall, this is the technique that is used. We can have this graphical result that we can download, and we can also take a look at a tabular result. So here we have a sense of our completeness and contamination values on the far right. I won't go into detail about all these different these 80s, 62 is the lowest. And this is just a reminder that we use DOS tool, and that performed an optimization approach using single copy gene markers. And so genomes that are very low completeness would have been chucked out here. And so that's exactly why we don't see any completeness scores below 60 in this analysis. Here we have a range of contamination values, and it's interesting to see things like this is thought to be 13.64% complete, which is kind of on the high end. So you might want to inspect this genome bin to make sure that there's no oddities there. Same with this one, that tends to be on the high end, and this one also. So while the completeness looks fantastic for a lot of these, and the contamination for a lot of these also looks great, it might be worth giving these bins another once over before swearing by the contents of their genomes. Okay, that was CheckM with sample A. Here's CheckM with sample B here, run like so. And here's a quick look at the results. Same marker gene plot as you just saw before, and relatively similar-ish results, I'd say. Good completeness, relatively good contamination. Yeah, actually contamination looks better in this instance, so that's interesting. Okay, if you're interested in downloading any of this CheckM information, both the plots and the raw data itself, you can click these links here. All right. We're doing pretty good. We're getting close to the end here. I may skip an app or two at the bottom to make sure we have time for questions, but we're doing all right. So now we're at step 7, which is to extract individual assemblies by domain. So at this step, what we've done is we're going to leverage the information produced by GTVB-TK to divide our bin contig objects into 2 different components, the bacterial and the archaeal components. And one note here, this is straight from Dylan's narrative, webinar narrative, which is a good note. The default is to extract all bins, and so we have to individually specify the bins that we want to extract. I'll show you how to do that. And then from this point, the other note is that from this point forward, and to streamline the presentation of this content, I made the decision to only focus on the B genome so that there's not so many parallel analyses. But oftentimes, you know, if you have multiple genomes, you're going to be running the same app multiple times with your different samples. Okay, let's take a look at the extract bins as assemblies from bin contigs object or app. So I input the location B bin contigs here. And as the note says, the default is to extract all bins. So it has that nice red text to warn you that that is the case. And so as I was prepping this app for running, I was moving through these genomes and looking at my GTDB TK results, and I essentially selected the genomes that all corresponded to the bacterial domain. And so that produced, that allows me to isolate this assembly set for just the bacteria. And so click run there, and it provides this result, which is nothing flashy to look at, but it just kind of gives a highlight of the operations that were completed. So it takes our genomes and it essentially rolls them together into this assembly set here with a unique identifier. The same operation was performed here again, but this time for the archaeal domain. And so I would have selected the inverse of the bins that were selected in this bacterial side of things. And I clicked run and got the appropriate result here and an assembly set that corresponds to the archaeal domain. So that brings us now to step 8. So here we are annotating the genomes. And so after using the DOS tool bin contig objects and dividing those up by domain, we are annotating them using the rapid annotation subsystem technology RAST pipeline to produce sets of annotated genomes. And one note that I have here is that the RAST tool, it provides separate options for annotating bacteria and archaea, and hence this is why we divided our genomes up by their domain. So this is how I ran RAST in the first instance. So the input here was the bacterial assembly set that we extracted at the previous step, and we gave it an appropriate name here. And then kind of folded in this advanced parameter set, there's this domain selection here where the default is correctly selected in this instance for bacteria, but that is changed in the next instance. And so here I would have clicked run, and it would have produced this result. So now we have, there's a lot of information here because essentially this is now an annotated, a set of annotated genomes. And so each of these entries contains information for the full feature, all the features that were annotated for each of these genomes. And so, you know, there's many thousands of genes that were called here. The high level, the nitty-gritty details are not as important as just emphasizing here that we obtained a set of annotated genomes, and they correspond to the bacterial side of things. Here we are doing the same operation, but the equivalent for the archaeal genome or assembly set that we extracted in the above set. And so this is where we diverged from using the default parameters. And so here we selected the archaeal domain and made sure that that was correct before running. And after clicking run, we obtained a similar result to the one we saw just before for the bacteria. We have thousands and thousands of annotated features for all of these different genomes. Okay, so now that we have annotated our genomes, we're at the final step in our process here, where we're going to build a tree and find relatives. So in the previous step, in the previous steps, we took our bacterial and archaeal assembly sets and pulled those out, and then we annotated those assembly sets to produce genome sets. And now that we have genome sets which are fully annotated, we can use the annotation information to perform a phylogenetic placement. And so to do that, we use this tool called an app called Insert Set of Genomes into Species Tree, and this provides an initial phylogenetic placement of our bins. Also sort of an independent glimpse into things compared to the GTDB TK tool. We also run a similar tool here called Build Microbial Species Tree, which constructs a tree with phylum level exemplars. Just a quick note, the NCBI RefSeq is used as the reference data for this operation, at least for the insert set of genomes into species tree app. And so that's something to keep in mind because it may or may not be ideal depending on your project goals or questions. In our case, we have a lot of divergent lineages that have not been cultivated before. And so we would expect RefSeq to have some blind spots if we're performing taxonomic identifications. Here here's a glimpse at the parameter set that we input. These were the extracted and annotated genome bins for the archaea. Sorry about that phone call. Here we kept, here I increased the number of public genomes all the way up to the maximum to give us the maximum information in our tree. This is by default at 10, so you may have to change that. And output from this tool are a tree as well as a genome set that includes your closest neighbors. So let's take a look at the result. This was for the archaeal domain. Okay, so our bins are, it looks like, intercalated within this tree here in various locations. We have this one up at the top, which doesn't tell us a lot of information because it's pretty distinct from other groups with known names. Here's the name here is bin 15. It looks like it's relatively related to this Aciduli profundum. So that's perhaps notable here. Also noteworthy and compared to the GTDB TK tool, here we have some phylogenetic context for our lineages. You know, we can sense the relatedness and the distances using this tree here, which is fantastic. Here we have, looks like a cluster of our archaeoglobi genomes. This looks like a fairly divergent archaeoglobi, which is consistent with a divergent archaeoglobi we published on and had some methane related genes, methane cycling related genes. Down here, it looks like we got a Thermococcus, which is pretty closely related to known Thermococcus. And here we step into a little bit more unknown territory. This is a pretty divergent group. Maybe this is the Hydrotherm Archaeota, or maybe this one is up here. Either way, these are fairly distinct genomes compared to the reference tree that we're putting in. So there you go. There's some phylogenetic context for our novel genomes. Here's the same thing for the bacterial side of things. We inputted the bacterial genome set here, increased the number of closest neighbors to 200, gave appropriate names to the trees and the genome sets produced. And here is the result. So bacteria, we have a little bit more coverage for them in the RefSeq database. So we expect that we'll have a bit more success identifying some of the closest neighbors here. And indeed we do. Looks like we have an alpha proteobacteria there. Acinetobacteria, all right, very closely related to this cultivar. Scrolling down here, Cupria vitis, okay, looks like a dead ringer for some proteobacteria. Here we go, here's some metabolic, or here's some phylogenetic diversity in our bacteria here. Doesn't look like there's very close relatives in the RefSeq database that we're using. This looks like an interesting placement within, I guess, Delta proteobacteria. I think that is intriguing and could warrant some follow-up. And then it looks like this group here at the bottom are fairly unique, and they don't have closest relatives in the RefSeq database. So cool, that's some interesting phylogenetic context that we have for our genome bins. Okay, that takes us to the Build Microbial Species Tree app, which is a new app that's still in beta. So if you see any bugs, please report them. But this is a cool tool that allows you to build a species tree in similar fashion that we just did, but this time you can include phylum exemplars so that you can contextualize your genomes, in this case, potentially novel phyla in the context of known phyla. So here we inputted the B sample. the bacterial side of things. And in this case, I selected the bacterial exemplar set, the one that includes isolates, megs, and sags, but there's a few other options here, including one that's both bacteria and archaea, one's that bacterial isolates only, and one that's archaea only. So that's how I ran the app. And let's take a quick look at the result. And so here are, it's hard to see, let me increase the window size. So here our bins are in yellow, and they're intercalated with these phylum exemplars that are listed in purple here. And so this is not a complete tree in the form that it's exhaustive and showing all the closest relatives, but this is a great tree in the form of it's highlighting how our lineages, our genome bins are fitting with what we know of current microbial phyla. And so to a first degree, we're getting the sense that, this is something relatively similar to the nitrospirae. This bin 10 here is something relatively similar to the acidobacteria. We got a couple bins here that look like they're members of the chloroflexi. Yeah, overall, I guess a very powerful tool, and these are hard trees to make in the form of it's not easy to know exactly what are the phylum exemplars to be included in an analysis like this. And so my colleague Dylan did a really good job of identifying thoroughly what lineages to include here. Anyone doing phylogenomics of novel lineages knows that making these trees and having them be complete is a major challenge. How are we doing on time? We only got 20 minutes left, so I guess I'm going to skip this second example with the archaea, but it essentially shows the same result from running this build microbial species tree. And that takes me to the summary and future directions now. So in this tutorial, we've showed you how to identify microbial genomes originating from raw sequencing reads, from metagenome sequencing reads. Specifically, this narrative tutorial, it highlights the advantages of using sort of a mixed binning approach with a variety of tools, and highlights how the end goal of using this approach can sometimes lead you to having the highest number of the largest number of high quality genomes. Once we had the high quality genomes in hand, we highlighted how you can investigate the taxonomic signal and identify novel microbial lineages. In the near future, KBase will continue expanding our metagenome workflows to provide users the ability to assess the taxonomic signal within raw sequencing reads using additional tools like Kraken and Metaflan. We're going to offer tools to potentially clean genome bins like RefineM, and we're going to offer additional tools to explore viruses and phages in your samples. With that, because I'm out of time, I'm not going to go into the unused portion of this narrative, and instead I will open it up for any questions.
Thanks, Sean. There's a couple of questions at the bottom of the Q&A document. It looks like there's just some coming in here, or if there's any that you feel like you want to give some more verbal response to.
Okay, so I'll start at the bottom of the Q&A document. How about that? Scrolling down. Okay. Yeah, first question, which looks like it's being answered in real time. Are there any SARS-CoV-2 specific tools that are being considered for KBase? Any timeline on that? Yep, the answer is dead on. Yes, we do have some tools that are targeted in this fashion, and that's really all that we can say at the moment. Is command line API available to access and run KBase apps and narrative? Dang, so that's a tricky one to answer. In theory, yes, there are some ways that you can use an API to access KBase apps and the narrative, but that is not the way it was designed. A big pro of KBase is having these things, these Jupyter notebooks, which are relatively navigable by all persons that are trying to look at these data. And so we find that retaining some sort of user interactive GUI really assists with the dissemination and communication of these data. There likely is an API for you to use if you're really insistent and would like to engage with our developers. Question, what is the practical implication of the app being in beta? So any of the KBase staff can correct me if I'm wrong about my answer here, but this answer, yeah, I guess it captures it. The apps in beta, they do have more bugs. The documentation tends to be incomplete. But overall, the app is probably somewhat running at that point. Like it's a safe bet that the developer working on the app is having some sort of success running that particular application. Otherwise, they would never promote it to beta. They would leave it in development. So I should, I guess, say there's 3 different stages. You can have an app that's in development, which is the first step. Then if you feel somewhat good about it and feel like it's functional, you can promote it to beta. And then once you've squeezed out as many bugs as you can muster and improve the documentation and tested it thoroughly, then it becomes a candidate for release in KBase. And so that's the comment on the process. All right, results from a narrative are maintained for how many days in KBase? Yes, forever. So a big advantage of KBase is that these data are there forever. So hopefully small labs, labs that do not have the overhead, the ability to pay for the overhead of maintaining servers long-term, they will see the benefit of bringing their data into a platform like KBase, where you will forever benefit from the DOE infrastructure and funding mechanisms to keep your data preserved. Question, can we take the input data directly without uploading from SRA? Yes, so this answer is correct. There is an import SRA as reads app that moves reads directly from SRA into your staging area, and then you have to import it from there. And so there is it's a 2-step process, but yes, that you can input your data sets directly. Next question I see here is, can we use all this approach for targeted metagenomic 16S sequencing? So let me just read this answer here. Yep, that looks great. Yeah, so this workflow was indeed focused on shotgun sequencing. And from within your bins, you can, within your shotgun assembly, you can look for 16S matches to determine what your mags match. But that said, 16S genes can be 100% identical, even if the rest of the genome is fairly different. And so if it's abundant in the amplicon sequencing and not abundant enough to make a good mag, it's probably correct. Yeah, I think another thing to keep in mind with respect to, I guess, just the topic of 16S gene presence absence is that metagenomes are not really good. Microbial metagenomes, when performing assembly on those, you don't often retrieve all of the 16S genes. In fact, you never retrieve all the 16S genes. That gene was selected because it has some conserved phylogenetic signal across all the domains of life, and therefore it makes it tricky to assemble. And so in your metagenome assemblies, 16S genes tend to drop out. And so because they're not in your assembly, they're also not going to be very often in your genome bins either. And so these are just things to keep in mind as you're moving through these data. Question about the webinar recording, and yep, this is recorded and you'll get a link. How do you determine if a bin has a high rate of contamination with the check-in table? Yeah, this answer is correct. It highlights that the last column in those tables corresponds to the contamination number. something to keep in mind as you're moving through completeness and contamination information. As your completeness goes down, your ability to detect contamination also goes down. And so what does this mean? It means that a genome that is, say, something like 60% complete, and as reported by CheckM, only say like 1% contaminated, it's likely that that 1% contamination is an underestimate because this is an incomplete genome.
Sean, there's a couple questions that were highlighted in blue that I think folks were not able to answer. If you scroll up.
Scroll up. Sounds good. Okay.
One's about diamond searches against which database to identify genes in the gene identification tool.
Got it. Okay, yeah, we'll do that one first. Diamond searches against which database? So Diamond, I don't think this, well, CheckM has an internal, or sorry, DOS tool has an internal reference database that of single copy marker genes that you download and run internally with this tool. And so Diamond is going to be searching for gene annotations in your data, and then it's going to try to make matches to those in the DOS tool database. So yeah, essentially it's the combination of the DOS tool database and your query genomes. Yes, and so the database that you downloaded is focused on single copy marker genes that DOS tool is using to perform its optimization step to maximize the completeness and minimize the contamination in your genomes. All right, this one above here, curious to know the total disk space occupied by the input files and results generated in this narrative. Yeah, it's a great question. So I had a bit of a snafu in that some internal development and reasons that are beyond this webinar, I had to rerun a bunch of these apps at last moment. And what I found was that for the apps that are heavily employing raw reads on system, they tend to produce very large numbers of and large size temp files. And so while in theory, you essentially have an unlimited disk space for your input files and your results generated in this narrative, as you're running all of these apps on system, sometimes you'll find yourself, you will find yourself working on a machine where the hard drive may fill up to 100% capacity. And so that's a bit of a segue, but you know, essentially there's always space for you to run your tools in KBase. If there isn't, let us know. And if you have disk space issues, please know that that's not uncommon and things that we often work through. Okay, those were the two blue ones. I'll keep scrolling down here to see if I can find anything new. This seems like a new question, so I guess I'll start here. Can we put all these apps in a single pipeline so that I just have to give a few inputs in the beginning and all the apps run in an automated fashion? This way I won't have to wait for the apps to finish. I'll just look over everything when it's completed. So yeah, good question. So we are slowly working on employing this type of approach where we have sort of predetermined, prescribed workflows for users to run their data from A to Z. But right now, that's still in development. For the time being, I think it's you have to kind of click through in the way that I presented the material today. And I think you know, part of the advantage here is that clicking through and having sort of checkpoints intermittently with your running of apps really helps kind of force your hand towards evaluating your results in real time and making sure that everything is running as expected. Yeah. Okay, can Kaiju and Gotcha2 be used for metatranscriptomic data? Yes, yeah, I believe they can. I've not used these for metatranscriptomic data, but as this answer highlights here, you can, and just make sure that you remove your ribosomal RNA component of your transcriptome library in no small part because Kaiju is a protein-based reference database, and so it won't work for our RNA. Question, what should we do with the genome that has high contamination? You mentioned we need to give it another run. Would you mind letting me know how we can do that? So currently our tools on system are underdeveloped from the perspective of giving you the ability to manually work through the contamination in your genomes. We're trying to improve this. As I mentioned, we're going to try to onboard a tool called RefineM, which helps to minimize contamination in your genome bins by pushing aside contigs that have aberrant coverage or tetranucleotide signatures, but those are not available yet. I can say that there's also interest in onboarding a package called Onvio, which is a very successful interactive tool, a Python-based tool that people are using to curate their genome bins successful. And I'll give an extra plug for Onvio in the form of, we would love to have it in KBase. It poses some additional sort of like broader challenges for us in the form of how do we track all of the real-time decisions that one makes when they're using a platform like Onvio that needs to be developed from the KBase perspective. But overall, Onvio is fantastic, and it was super useful for me to clean up these genomes for their original publication several years ago. So check that tool out if you have not already. I don't see any other questions populating at the bottom of this document. I'm happy
Awesome.
to, yeah.
Thanks, Sean. I think we can wrap it up there. Thanks again, everyone who stuck around for this. We'll be sending around the video as soon as possible. But yeah, thanks again, and look forward to some more KBase webinars coming soon.
Thank you.
Bye-bye.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOEKBase. And if you have questions or encounter an error when using KBase, please contact the help desk.